Hello, welcome back. We'll have a presentation from Professor Marcello Massimini from Milano, but it will be through Skype, I was told. I hope everything works. We can try to get the contact and... Oh, hello Marcello.
Can you see me?
But
Hello?
Hello Marcello. First of all, thank you so much for being there.
Thank you, Stephen.
So I think we're all
yeah.
set, and the floor is yours to talk about integrated consciousness research within HBP.
Thank you, Stephen. Everybody, apologize for not being there. I got a lung infection and I've been forbidden to travel, so I'm doing this through Skype. And so just to give you an idea of the state of your speaker, a close-up now, this is how I look like. And it feels actually quite weird to give a talk to such a top international conference with the best scientists and philosophers from my my bedroom. It's even more surreal if you have a little bit of fever, but I'll do my best really. And I hope technically everything works. And please give me feedback if the audio doesn't come through properly or the video also is not good. So my task here is to give an overview of the consciousness relevant research that is ongoing in HBP. You have heard and you will hear from other researchers that have much more experience than me, longer experience in the project. Katinka, Jean-Pierre, you will hear from Cyriel Pennartz, Alain Destexhe, so I will not overlap with them. But actually, I'm relatively newcomer in the project because I came in about 2 years ago. So maybe I'm in a good position to give a flavor of what's going on in the project that might be of interest from a perspective of an outsider. So I'll try to do this, and I will start from this graph actually that I often use. This is a simple graph from Christoph Koch and Giulio Tononi. And it makes a very simple point actually. That is, we assume that there is a relationship between function, intelligent behavior by an input-output perspective, and consciousness. So basically, if say an animal shows flexible, complex input-output behavior, we assume that he has, high capacity for consciousness. And this might hold true generally in nature, although already might be problematic to compare on this graph, say, an octopus and a cat. I wouldn't know where to put them, but it might definitely become problematic in the future, because we are actually manipulating and changing nature. And so in one direction, we are building machines that are very fast. They might outperform us in many tasks. For now, only specific tasks, but they do that. And so they're very good in terms of intelligence, input and output function, but they're not conscious, presumably. On the other hand, we are intensive care medicine is saving hundreds of thousands of patients from severe brain injury. These are new brains that would have survived until 50 years ago. And sometimes what's left is a brain island or an archipelago of brain islands. This is a figure from a famous paper from Nico Schiff. You will hear more about this from Stephen, Olivia, and Nico later in the meeting. But this is just to say that we also have, not only we might have intelligence without consciousness, but also consciousness with no input and output function. And so consciousness would score basically almost 0 on the x-axis of this graph. So things are going to get confused in the future, probably, and we need to be ready. And I think society already feels that. It is enough to see, how many movies have touched upon this problem, successful movies in the recent years. This is Eva from Ex Machina, but then we have Samantha from Her, and then we have a Blade Runner, and all movies dealing with the problem of disentangling between consciousness and intelligence, if you want. And this is really a serious problem. And actually, myself, I was really impressed, like many of you, when I saw this. And this became very famous, AlphaGo, then AlphaZero. It was impressive, even for somebody who is in the field, to see this happening. Although I know this is not general intelligence, but it's sort of striking. And I can see also this confusion in my family, if you want, because my two kids, one is 6 years old, the other one is 8 years old. They play with the iPad. Sometimes they talk to Siri. And sometimes I saw them actually confused between Siri and their cute little cousin Julia. She's 6 months old. Actually, they were making fun of her because Siri was answering all their questions, and Julia was basically answering to any question, ta 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 ta, papa, and they were making fun of her. So this is, of course, not much of a problem, but I can see, and maybe Katinka is much more of an expert on these aspects, also an educational issue in disentangling and distinguishing between function, intelligence, and experience and consciousness in the future. So now, what about HBP? I think HBP is in a privileged position to address many of the problems that are, represented in this graph. And I give you just, an example, because I was looking at the representative figure from HBP, and I took this figure from. So I think we were left pretty much here with this graph showing the potential confusions between intelligence, consciousness, function, and experience between, with the development of artificial intelligence, brain islands, patients that are disconnected from the external environment, and how HBP can fit into this or contribute to this problem. I was showing this figure, and this figure is taken from a paper by Catherine Ammons, Neuron, 2016, and it is basically the manifesto figure of HBP right now, and it shows that this project goes across scales, species, and models, from molecules to systems, but also it shows clearly that the project also addresses, includes at the same time experimental neuroscience, including clinical neuroscience of brain injured patients. And you will see that with Stephen and Olivia. And also at the same time, and this is sort of unique, cutting edge simulation, brain inspired artificial intelligence, and robotics. So it's really in a position to explore the entire span of this graph between intelligence, consciousness, and all the possible dissociation between this. So I think that this is a good starting point if you put, things in this perspective, and addressing the problem of the relationship between intelligence and consciousness and the potential dissociation. I'd like to start from something that is very present in HBP, that is the simultaneous exploration of the cerebral cortex and the cerebellum. And this is, again, it's a unique, because there is no project where you have both at the same time. And I think that this is important for many reasons. So let's take the cerebellum, for instance. The cerebellum is a beautiful piece of machinery, extremely smart. It builds detailed models of the world, it has a fantastic predictive power. It helps us moving about the world smoothly. If I'm speaking now and I roughly just have a rough idea of what I'm going to say, is thanks to my cerebellum. If I can ski negotiated bumps in the snow while admiring the peaks of the mountains, it is thanks to the cerebellum. If I can drive a car not only in Stockholm, but also in Naples, maybe when I'm talking to my passenger, which is very challenging. And I would challenge a Tesla or Google car to do that. It's largely thanks to the cerebellum. And also, if sometimes out of the blue, I have an intuition and a solution to a problem, it is probably thanks to the cerebellum. It is definitely a very smart piece of machinery. And also, there was this interesting paper, I don't know if you saw that, in a scientific report this year, showing that one of the reasons, or it's an hypothesis, why Homo sapiens survived to Neanderthal seems to be that he had a larger cerebellum. So it's really, a remarkable piece of machinery, functionally speaking, but also as a physical object is remarkable. It has, it's small in size, but it's very big in numbers. It has 4 times more neurons than the cortex, about 80 billion, roughly, neurons. It's a lot, really. It has access to all kinds of sensory inputs. It affects all kinds of motor outputs. It has inhibitory, excitatory cells, neurotransmitters, neuromodulators. It's really remarkable also in this respect, if you compare it to the thalamocortical system. But it has a problem that is, it seems not to be conscious. We know that because whatever the cerebellum learns goes below the radar of consciousness. And also we know this because when you remove the cerebellum from the skull of a patient, maybe because of a tumor or because your other reasons, the patient walks away, maybe clumsy, but is vividly conscious and intensely conscious, just as before. So the cerebellum, it is very, very different from the cortex in this respect. And it's really interesting now to understand why. Okay, we know things about it. We know that the cortex is more like a core of linked by recurrent connections that are both specific and integrated, while the cerebellum seems to be a feed-forward network. And this is already very interesting. If you think about it, the cerebellum receives about 200 million inputs through the mossy fibers, and those diverges into 50 billion granule cells. It's like the hidden layer of a deep learning network, huge deep hidden layer, and then it all converges on the Purkinje cells, and they receive each of them receive about 100,000 input, and also a powerful error signal from the climbing fiber. So it really looks like an artificial neural network. It's all feed forward. It's extremely fast, extremely plastic, extremely adaptive. And this is quite interesting, I think, to explore. This is really something that we can do in HBP. In HBP, we have detailed simulations of the thalamocortical system. Here you see a very realistic biophysical simulation, detailed biophysical simulation of cortical neurons. At the same time, in the same project, and this has been done in Pavia, and it's unique. We have a detailed simulation with all the neuronal compartments of the cerebellum. This is a Purkinje cell firing, and you can compare it to what happens in the cortex, more or less. In a way, we will be able to actually really see how the structures are working, not only at the level of single neurons, but also at the level of network activity. Again, you see on the left side this beautiful like fireworks produced by a real simulation of cortical neurons. This is courtesy of Sean Hill in APFL. And on the right side instead, you see this sort of constellation of neurons becoming active. So here you see Golgi cells, the yellow one, and granule cells that are the most numerous cells in our skull. And this is really a simulation of the dynamics. And comparing these two universes that are so similar in many ways, but very different when it comes to consciousness. One is very, alive and hot, and the other one is pretty cold in terms of consciousness, the cerebellum. It is very important. And this can be done through simulation, especially because the cerebellum is very difficult to access because of its position, and it's also very difficult to record because granule cells, they are the most numerous cells in our brain, but also they are the smallest, so it's very difficult to record from them directly. So obviously simulation will play a major role here, and we'll have many, many things to learn, I think, from this comparison in terms of, why systems can be intelligent but not conscious, and what are the key differences between these structures. Also, I think that exploring these differences is important in more general terms. And now I'm going back to actually another graph. Oh, by the way, they are tightly connected, this universe. Very, very well connected, but still so different. Now I move to this figure. This is a figure again taken from Christoph Koch and Giulio Tononi, and again I think comparing the cerebellum and thalamocortical system might be helpful in general terms and in the light of this figure. This figure shows that there are different ways you can implement the same input-output relationship. So you can do it with a complex integrated system on the left side, which is hard to do. It's very complex to build, difficult to build. Or you can just unwrap this system into a purely feed-forward system by exploding the number of elements, and you obtain the same input and output. So by an extrinsic perspective, functionalistic perspective, these two systems are really equivalent, but if you look inside, they're very different. And now the question, which is relevant in general, is does it make a difference for consciousness? How they are made inside, the kind of interactions that are hidden inside, above and beyond input and output. So if we go back, if we take the example of the cerebellum and the thalamocortical system, it seems that it might matter a lot, that we need to look well beyond the extrinsic perspective of input and output and go look inside to how things are, how elements are interacting, to the kind of causal interactions, the strength, the cause-effect power intrinsic to the system. So this probably it's something that we will need to do, even because, and this brings us back to the initial figure slide where you compare the feed-forward artificial intelligence system and the brain island in the patient. We will need to find ways to go beyond this extrinsic perspective and look at the internal relationship, even because not only we are saving patients with brain islands, but also we are growing brain islands that are disconnected from input and output in a dish. For now, they are very small, but soon we'll be able to grow three-dimensional islands and organoids, and we will need to know, or at least have an idea or a criterion to decide when to stop and how to deal with that. Also, and this is actually happening within HBP, we are building neuromorphic chips, and this is different from a simulation. This is actually a physical implementation of neurons. So physical, biophysical properties, passive, active membrane conductances are replicated, and it's all happening physically on a chip. And there are connection, there is plasticity. This is a wafer from brain scales. This is done in Heidelberg by Karl-Heinz Meyer. It's about 200,000 neurons and 44 million synapses on one wafer, but then Karl-Heinz Meyer is also connecting this together. And, you know, this amounts to about 4 million neurons and, you know, a significant number of synapses. You have spike timing dependent plasticity. And also you could have recurrent connections. And the thing is that these artificial neurons can be 10 times faster than biological neurons. So they are able to learn. They could be exposed to an artificial environment, say virtual reality or internet, whatever. And in principle, they could grow, especially if they are put under constraint of space and number of elements of energy, they could grow a considerable amount of complexity. And also here, we should ask the question, does it matter what kind of internal causal interactions are we growing? Is this feed forward? Is this recurrent? Is it a complex system? We might probably be called to answer these questions. So to look really at the internal interaction. So here, you know, there are a few interesting things happening within the project that I want to point out. So the first I didn't even know about, and I found it really, really interesting, is the work by the group of Idan Segev. This is a work by Eyal on eLife 2016. It's quite impressive to me, at least. So it's about difference between the neurons, cortical neurons, in rats, in rodents, and humans. And it's a very interesting work that, again, shows the power of combining experiments and simulation. And basically what they did here is that they took fresh cortical tissue from humans undergoing surgery for epilepsy. They recorded from the neurons the properties, the basic electrophysiological properties of the neurons by injecting current. They also reconstruct the morphology. Then, based on this information, they built a model. And then the best model, the best fitting of the data, the model said, was a capacitance of the membrane of about 0.5, which was really odd and different from the typical capacitance that is used in modeling, and it's derived from rodent data, that is 1 microfarad. So it's 0.5 instead of 1. So they were curious about it, and they went on and they isolated cortical neurons from humans and from rats, and they actually measured the capacitance to test this prediction of the model. And actually the experimental data shows that there is a substantial difference in capacitance between the mouse and the human, 0.4, 0.5 in humans, about 1 in the mouse. And this has a huge impact on cause-effect relationship between neurons, on the cause-effect power internal to the system, because, first of all, if you have a lower capacitance, and with these values, you have EPSPs that are larger by between 80 and 90% on average. Also, you have you need less synaptic input to generate a spike, and this is again a substantial difference. So with low capacitance, 100 excitatory inputs are enough to fire versus 170. This is again from simulation based on the data. This is substantial, and in a network might make a huge, huge difference. So above and beyond, you know, brain morphology, number of neurons, arrangement, networks, it is important to also evaluate these intrinsic properties, and they might make a huge difference from the internal perspective, the intrinsic perspective of the system. Another example of this kind of exploration in HBP is the work by Matthew Larcombe. And this I find, again, very, very interesting. So we know that we have feed-forward sensory input coming from the external environment, impinging on middle layer of the cortex, while corticocortical feedback connections are actually connecting to the dendritic tuft in the superficial layer. And this is already quite strange, because one would say, well, these inputs, corticocortical inputs, are very important. They deliver the context. They must be relevant. But why do they get there where, you know, they're so far from the soma, and it is so difficult to generate a spike in principle? The answer is this very important discovery of apical amplification. Basically, the fact that there is a zone in the apical dendrites where calcium spike can be generated, and these calcium spike are more likely to be generated when at the same time you have a back propagation from the soma. So basically, due to this mechanism, it is not enough to receive an input to the surface surface layer through corticocortical connection. It is not enough to receive a feed-forward input to the soma, but because in the first case you only have a small depolarization, in the second case you will have a spike, but if you get both, you will have this calcium spike, which triggers a burst of action potential, a powerful burst of action potential, which makes a huge difference. Basically, this is really the coincidence between this feedback and feed-forward connection is driving powerfully the neuron. This has been postulated as a fundamental mechanism of matching external data with internal prediction, and it's a beautiful model, actually. And by the way, within HBP, this layer-specific contribution to sensory perception has been also explored in humans using 7 Tesla fMRI. This is work by Lars Muckli, where they show that when you have a visual occlusion paradigm, the contribution to of superficial layer is crucial. So this feedback coming to the superficial layer is basically filling up the occlusion, and this is very important. And of course, I mean, there is the work by recent work by Matthew Larkin, and I don't know if you maybe
the cerebral cortex is made up mostly of tall cells that look a bit like trees. In fact, the extensions coming out of the cell bodies are called dendrites. That's ancient Greek for trees. We've been studying their properties and hypothesized that a hotspot in the dendrite.
So I don't know if I'm confused whether you have an audio or not, but anyways, this work makes the prediction that a calcium spike should happen at the threshold between subliminal and supraliminal, where you have the detection of a stimulus. And actually, this is what happens.
Right. Might be activated around the perceptual threshold. To look at this, we stimulated the whiskers of the mouse while using a genetic technique that allowed us to visualize dendritic activity under a 2-photon microscope that can look into the brain in real time. We found, as predicted, that the hotspot fired exactly when the animal reacted to the stimulus. But even more importantly, we found that suppressing the hotspot increased the perceptual threshold. That is, the animal became worse at perceiving the stimulus. And on the other hand, activating the hotspot artificially lowered the threshold, making the animal more sensitive to the stimulus. This leads us to the conclusion that the hotspot in the dendrites of cortical neurons is somehow crucial for perception.
Okay, it's me. Sorry, I closed the conversation by mistake. I'm sorry. I'll start now, right now. Okay, thank you for being so patient. So this, in the words of Matthew Larkin, much better than mine right now, because I'm not really in great shape, I have to say. But so, and you will hear also how feedback is important. There will be a talk later on by Pieter Roelfsema on a much larger scale, though, in monkeys. I just want to add here that this mechanism of dendritic amplification is modulated, can be modulated by anesthetics, by GABA B and GABA A. Can be suppressed by GABA B, GABA A, can be suppressed by Martinotti cells. There are plenty of interesting modulation. It's also enhanced by adrenergic modulation. So it's obviously relevant for conscious detection, perception, but might not be necessary for conscious per se, because actually this apical amplification is not present during REM sleep, but we still dream, although these dreams are clearly disconnected from the outside feed-forward sensory stream, and this is very interesting. And also, it comes rather late in development. So this leaves some question open, and it's very interesting to explore that, but also suggests that above and beyond sensory input, we should look into the brain also directly, and to look into internal interaction above and beyond sensory inputs. And this can be done, and we've been trying to do this within the project with TMS-EEG by developing this measure of internal causal complexity. So again, by an intrinsic perspective, we measure the complexity of internal interactions by zipping the deterministic response to a perturbation. And this works quite well in benchmark conditions when subjects are conscious or unconscious. They report being conscious or unconscious, whether anesthetized, dreaming, sleeping, or whatever. And also, it might be useful at the bedside of patients. And if you stratify patients, actually, disorders of consciousness, you see that you have a very good sensitivity in minimally conscious patients. So they all score in the complexity range typical of the conscious benchmark. But also you have an interesting stratification of unresponsive wakefulness syndrome patients. So basically you have three ways, by the TMS-EEG point of view of complexity, three ways of being UWS. So the first way is, well, not very interesting, is no response, no causal interaction whatsoever. But this is actually happening in post-anoxic patients, and probably reflects the fact that the cortex is destroyed and completely unresponsive. But then there is an interesting second way that is a sleep-like, low complexity way. And then a third way where you have high complexity, levels that are comparable to the ones of the conscious benchmark. And this can happen in patients who are disconnected for different reasons. We had different cases, interesting cases. This is just one. This patient had a midbrain injury plus an hemispheric injury, and he was temporarily completely disconnected also from the sensory side. And then, but he had high complexity, and then the patient recovered after a few weeks. And so there is a way to measure internal complexity above and beyond input and output that might be useful to assess patient. Of course, this is very complicated. So within the project, there is also a very active exploration of simpler metrics of consciousness that are based on spontaneous EEG. So this is work with measures of effective connectivity based on spontaneous EEG done in Oslo. On the right side, there is a comparison between this perturbational complexity on the y-axis and multivariate brain machine learning based classification of EEG in patients. And these are very interesting. The goal is to get the best out of it, to combine this measure, see where they differ, and basically get the best to use them to be used at the patient bedside. And also there is a comparison between structure and complexity. And it's interesting to notice that, yes, brain complexity in terms of TMS-EEG responses can be partially explained by the degree of injury, so by structure connectivity, but not completely, because, for instance, you have patients who are structurally intact, metabolically active, but stuck in a state of low complexity, sleep-like sort of low complexity. And then you have other patients that gross connectivity being equal, recover complexity and consciousness in the space of a few weeks. So there must be some functional balance, some change in the circuit that occurs that changes the properties crucially in the space of because of some functional happening. And so we need to explore that. And actually, in the project, in HBP, there is an active line to understand how complexity grows and collapses in the thalamocortical system, and to connect complexity to slow waves. And actually, the hypothesis is that complexity collapses, structure being equal, for instance, when we fall asleep, because neuronal properties are changing. Like, for instance, neurons are becoming bistable, and they tend to enter a down state after initial activation. This is also the basic mechanism of the slow oscillation, and it is due to calcium-dependent, activity-dependent potassium currents. It might be due to excess inhibition, but basically what happens is that neurons are receiving an input, then they shut down and they recover activity stochastically. And this would be really a killer for complexity. And within HBP, we are exploring at multiple scales if this is actually the case, because of course you cannot tell from the scalp. And so for this reason, we are combining, for instance, intracranial stimulation and recordings and scalp EEG recordings. And this is an example. This is what happens when you are stimulating the brain with TMS and you record the EEG response in wakefulness. This is what happens in sleep. And then we can replicate this basically intracranially. And I will show you the movie here. So this is what happens in wakefulness when you give an intracranial stimulation and you record the EEG response from the scalp and intracranially. At the same time, you see this complex pattern of activation. And then this is what happens in sleep. So you have the same exact stimulation, but a pattern that is stereotypical and local, so complexity is lost. But here you can look at the local field potential and try to understand what are the neuronal underpinning of this loss of complexity. And indeed, this kind of exploration between the macro scale and the mesoscale shows that when complexity is collapsing in non-REM sleep, this is because neurons are actually becoming active at the beginning, and then they go into a down state as reflected in a suppression of high frequency here in this time frequency plot. So actually there seems to be this simple neuronal mechanism behind loss of complexity, at least during sleep, which breaks down causal interaction between neurons. And this is also explored in Oslo in rodents. And it's very interesting because we can replicate the same, we can have a measurement at the bedside with TMS-EEG at the macro scale. Then we can link the macro scale to the mesoscale through intracranial recordings in humans. And then we can do the same intracranial stimulation and recordings in rodents. And this is what happens in wakefulness in a rodent. This is work by Alessandro Arena, Johan Storm in Oslo. So you see this complex pattern of spatiotemporal activation in the cortex. And this is propofol anesthesia. Actually, it is very similar to propofol anesthesia in humans, but also to sleep, and you see the complexity drops dramatically, and so the spatiotemporal dynamics. And again, you can link this loss of complexity to this simple neuronal mechanism that is a neuronal downstate, that is neuronal bistability. And finally, with MAVI, we've been exploring this in slices. Slices are nice and very useful, I think, because they sort of represent an extreme case of cortical island. Second, because you can do any kind of manipulation on a slice, pharmacological, electrical, you have full access to the slice. And actually, it was important for us to adapt this measure of complexity to the slice to show that complexity is low when the slice is in a resting sleep-like state, that we can manipulate complexity in some ways, but not in other ways. So that by, for instance, adding noradrenaline and carbachol to the bath, you have an increase in complexity. You basically wake up the slice, and you have this increase in complexity in this small network, but it is not enough instead to increase excitability. So this gives some, ideas and some indication on how we can possibly manipulate complexity in the future. This is just a starting point, but of course it's the beginning of a multi-scale exploration, which is typical of HBP, of this mechanism of loss and recovery complexity, which will take advantage of simulation, computer simulation at different scales. I think this is relevant even because in some vegetative state patients, we see the same thing. So we see the loss of complexity, downstate, and then when they recover, we have a growth in complexity and the downstate disappears, basically. So exploring this on a multi-scale, using simulation to make prediction, devise new treatment might be interesting at least. Then I'm done. Basically, this is my last slide. And I just want to go back to the initial point. So I think, yes, we can have definitely experience and consciousness with no computation, no intelligent behavior or input and output. These are two examples, quite nice. One is from a patient who is actually just recovering from mild traumatic brain injury, car accident, and another patient who's recovering from a syncope. This is a first-person report about how it feels, this pure sense of presence. And on the other hand, we are, getting closer to have systems that are very smart, very intelligent, and unconscious. And also, sometimes in the future, we might have we might happen to build systems that are intelligent and conscious. For instance, I don't know. This is an open discussion, but what if a neuromorphic computer with billions and billions of neurons develops very high levels of complexity and is also very functional by an input-output point of view? We'll have to discuss that. But of course, this is a complicated landscape, and I think that HBP can contribute to this landscape. And actually, I think it's a landscape that it's quite interesting, and I am just looking forward to hear the discussion about this. Thank you very much. Thank you very much for your patience. I know it's been a very suboptimal talk. You've been very kind to me, and many apologies. Thank you.
Okay, so we start this session then. The first speaker will be Cyriel Pennartz. He is a professor of cognitive and systems neuroscience at the University of Amsterdam, and he's a partner of the Human Brain Project. And today I found out, apart from having done quite a lot of work on neuroscience in different areas, he also studied philosophy, and he's going to present next. Thanks, Cyriel.
Thanks, Mavi, and luckily we're not totally dependent on Siri to make an intelligent decision about the non-functional Skype connection. Okay, so we're going to rapidly switch after this to more the experimental neuroscience, but I'll also address some theoretical issues in connecting to the speakers this morning. So I'll address also animal consciousness. What can we say about it theoretically and experimentally? So if I sort of try to outline where I place consciousness, then it's kind of difficult, as you might realize, to really define consciousness head-on. We can say we lose it when we fall asleep, we regain it when we wake up, but then, or when we dream, then one has to define those terms like what is waking up, etc. So there's a problem of circularity, that we always have to rely on similar concepts like experience, subjective experience, etc. So instead of this head-on approach, one can ask, well, you know, if it's so difficult to define consciousness, then why is it so easy to answer the question, are you conscious now? And probably most of you would agree, and I hope that you're still conscious and not falling asleep. So apparently that's very easy, but why is that so easy to talk about your own conscious being conscious or your next door neighbor? So that's certainly an interesting question to address, that we also meet up with animals, but also these days, indeed, AIs and robots. So instead we could ask, well, what are the hallmarks of consciousness? What do we find reasonable to take as indicators of consciousness? First, subjectively, I'm going to run through this quite quickly because there's some familiar points. I think qualitative richness in subjective experience is a hallmark. By that I mean it's multimodal, it comes from different senses. The senses have submodalities, like at vision we have motion, color, etc. But also immersiveness, it's an important property because it's not just here that we look at the Caravaggio painting and sort of have purely a visual experience. It's also that we're in a situation to experience that, which in this case is being in the church of the San Luigi in Rome, having a certain body position. You hear murmuring all around you, clicking of iPhones, etc. So with the smell and everything, it's a highly multimodal experience. But we're also in a situation to have that. So we're not looking at a video screen as if we're playing a video game. We're sort of in the middle of the scene as we experience it. Second property being integration, that already has come to pass. Here I illustrate this by the attribution of sounds to the same lips that are moving. So of the puppet by this ventriloquist, there's a certain unity to it. There's also a certain dynamics and stability to perception. That is, we can flip between the front and back sides of the Necker cube and make it switch from lower right to the upper left corner, but this can only happen at a certain speed. It's not at an arbitrarily high rate. So we also have a certain stability to our perceptions. There's dynamics that can be explained in terms of the underlying neuronal dynamics. First, a person perspective, the thing that we have, our visual experience disappearing, but no, the fact that we have one common viewpoint, also a body-centered experience is important here. I don't know why it fails to move on, but I'll just keep on clicking patiently. It's another... there we go. All right. Last thing to note is interpretation or intentionality. The fact that we do have a brain that is somehow able to tell us things that are about something else than themselves. So neuronal activity can somehow be related to vegetables and fruits without having those vegetables and fruits in the brain. So I think this also refers to the projective capacity that Jean-Pierre Changeux mentioned. Now, to study this in animals is not so easy because we don't have the same kind of subjective access that we have. But I would make the case that indeed many animals, particularly vertebrates, mammals, can sustain conscious states due to several arguments. One is the similarity in electrophysiological brain states. What you see over here is the comparison of human sleep states like REM, non-REM, etc., with mice or rats having very similar physiology in terms of not so much the duration of the sleep states, but the EEG markers that we attach to non-REM sleep and REM sleep, such as hippocampal theta rhythm, etc. Similarity in the brain anatomy is another indication, but also behavioral markers that are important. Basically, one could say there are basic markers that could indicate that something is going on. I wouldn't say it's sufficient, so like sleep-wake states or behavioral reactivity, like orienting responses, can be taken as a basic marker, but it's hardly going to be sufficient. There are more advanced markers to consider here in animals, one being the similarity of animal psychometrics with human psychometrics, in this case illustrated by a visual contrast curve in mice. I'll talk about this a bit later on. where you see basically that a higher visual contrast leads to more detection responses. And also, when you add an auditory stimulus in this visual detection task, you see that the psychometric curve is shifting towards the left. It becomes steeper. This is more or less exactly like in humans or primates, for instance, but the contrast sensitivity is actually quite high in mice. They more suffer from a lower visual acuity. We have different markers in the advanced sense. We could say there are, for instance, responses to illusion inducers, like we have over here in cats. You see the rotating snakes illusion on the right over here. It's quite small, so you might not have this experience, but the cat is certainly acting as if it's reacting to something, yeah, sort of crawling there, or you might say it's trying to catch something. And that is very, I think very indicative of having some kind of experience there. We have additional indications, for instance, from the use of episodic memory in animals, which is linked to consciousness, and this is where you also find it back in the textbooks, because episodic memory or declarative memory is defined as having conscious recall of experiences. So when we see indeed indications that animals use episodic memory in the sense of combining spatial information with temporal information, with object information, that can also be indicative of consciousness. And then there's goal-directed or planned behavior that I'll talk about next. Interesting is the recent addition, and I was hoping that Marcello could have addressed it already, which are theory-based measures of consciousness, such as perturbation complexity or other measures of complexity that could help us out when we have to estimate consciousness in non-responsive patients, for instance. But it could also work for animals and other beings. There can also be additional measures like representational measures, like how much representational content can be represented. What is the information carried and the information transferred between areas? Should we draw a line there? Should we say, well, some animals or organisms are not conscious, and why not? How should we do that? Here you see the behavior of Paramecium. It's a one-celled organism that is quite seemingly adaptively responding to its environment. It has cilia, and these cilia are pressing against objects. So then it will sort of curve around or retract and take a different angle. But one could argue here that this is purely a sensorial behavioral reactivity that is purely explainable from the mechanosensitive properties of the cilia and the way they cooperate to steer the organism in another direction. So here I would argue there's no need to suppose anything like perception or imagery or dreaming as the prime examples of consciousness. Yeah, and then, in relating to theories like IIT, for instance, it's important to address that it should be important to have manifest properties of consciousness in some way that are verifiable and observable apart from the conditions or predictions that the theories make. Let me illustrate what I mean. So on the right hand, we have a knocked out boxing person, as over here, where you clearly see a distinction from consciousness and non-conscious states in the same person, basically. And this is typically a hallmark that whenever we call something or somebody conscious, there are typically also non-conscious states. It's not that they are similarly conscious all the time, or just a little bit conscious sometimes. On the other hand, the smoke detector here is reactive. It has a sensor. It senses smoke, and it will sound an alarm bell, but there's nothing in the way of that we need to suppose consciousness with some kind of manifest property. Compare the situation to physics, where it's also very natural to ask for or demand manifest properties, like observable properties of ice, of a frozen state, similar to a conscious state, and that as opposed to liquids. So ice has solidity. It has a crystallization form that is measurable and observable. Whereas on the right-hand side, you see the result of a Belousov-Sabotinsky reaction, a chemical reaction that results in chemical compound mixing and reacting, and they generate a complex evolving pattern, which has a certain integration and differentiation. However, it does not have this manifest other properties of consciousness that we need basically to verify. And I would argue that such systems are thereby not conscious. It's not reasonable to call these kind of systems conscious, and therefore the consequences also be that panpsychism is not an option. Theories of consciousness in the end should be able to discriminate conscious versus non-conscious states and also allow means to verify that this is the case. In which kind of biological settings do animals need consciousness, or humans? So here is a kind of functional take on the problem of consciousness, which means, again, that we can say reflexes, for instance, do not need to rely on consciousness, and the reason is that you just need to make a very fast response to a simple stimulus, like retracting your hand from the electromagnetic heater over here. It is interesting that awareness follows that reflex response, and we can talk about why that does result in awareness of pain, etc. I think that's highly adaptive and something we learn from. The other hand of behavior is goal-directedness. So the contrast is basically I'm making is between reflexes and habits on the one hand, so simple stimulus response behaviors versus goal-directed behavior. That is not simply behavior that is driving you towards a goal, but rather behavior that depends on a representation of the goal before you undertake the action. It's a notion well worked out in animal and experimental psychology as well by Anthony Dickinson and colleagues. So this refers to having a plan before you undertake an action. How do goals become selected is an important question. Well, you need, in addition to homeostatic needs, which you need to identify from your body state, you also need to know what your situation is like in order to select the most appropriate action. So what we here see is that, let's say, the biological function of consciousness is basically to support this decision process by providing a whole survey of your situation, including your own body. And this is meant to be illustrated also by the orcas. It's a little movie. And the orcas here, you see how they collaborate socially to try to capture this sea lion, which is hiding or trying to escape on this ice shelf. What they basically do is to generate waves, as you see again over here, collectively to try to tip over the sea lion of the ice shelf. But this requires, I would argue, a very accurate world representation. Like, where are they themselves? Where are their conspecifics? What is the physics of the situation? Where is the sea lion supposed to be? That, I would argue, is the kind of behavior that requires consciousness, at least in the sense of generating an immersive survey of the situation and the body. On the other hand, you could argue, well, could this not be done by artificial creatures? Well, perhaps it can. If they work purely digitally, that wouldn't work very well for a biological creature. Just having lots of zeros and ones, you would have to identify what are they about. So it makes sense to build a sort of intermediate to fast qualitative representation of the scene. So what are the facilities to enable deliberative action? I would argue that is this conscious representation plus episodic memory. A little bit of theory of how perceptual experience might come to be rich. And this might seem to some as a trivial matter because our nervous systems and sensory organs have lots of feature detectors in the skin, also by vision, etc. A theoretical problem, however, is that, well, there's the labeled lines hypothesis that we know from Johannes Müller, and that says, well, what we come to experience purely depends on the nerve fibers that transmit the information from the sensory organs, so that's all specified. The problem, however, is that from the brain's viewpoint, those inputs are unlabeled. They arrive not as in the manner depicted over here, where you have action potentials flowing into the brain, and then when they arrive in the visual cortex, the information becomes visual. This cannot be the case like that. It's not a good way there. So the labeling, actually the identification of the modality is lacking, and it has to be somewhere done centrally. Also, the way we think about classical neural nets is not that they have a capacity to identify their inputs or outputs. It's basically all Chinese, or, well, supposing you don't know Chinese. So a couple of predictions from this theoretical reasoning is that the sensory modalities actually collaborate. They have to talk to each other, not only to process the individual specific inputs that are unique, but they also have to interact to establish basically their unicity as a relational property. That is to say that the qualitative properties have to rest on a sort of high-level network property which, for instance, refers to the coherence or correlation that we have between vision and audition or proprioception. This is more or less the psychometrics of such interaction. At the anatomical level, however, we have to say that instead of talking about an area that is intrinsically visual cortex, we rather have to say, well, there's some input there that transmits information from the LGN and from the retina, but to eventually end up with vision as distinct from audition, taste and smell and so on, there's the interaction going on with many areas to build a reasonable world explanation. One prediction is that these relationships are to be quantified by correlations and decorrelations. One can also capture it by saying that they have predictive relationships to one another. So some modalities predict other modalities better. One of the predictions for experimental neuroscience being that primary sensory areas, but also other visual areas, for instance, will not be purely unimodal. That is, they might as well interact and process information from other senses. A final theoretical note relates to the heart problem, actually. We argued that a possible way out is to create a non-reductive framework, to say, well, we have a visual hierarchy over here where information is being propagated. Usually, well, people might say, well, the interpretation or inference that is going on in perception has to happen at a higher level, so prefrontal, parietal, etc. But if you just look at the physiology, this would mean that the only thing you find at higher levels are more spike patterns, synaptic inputs, transmitters, etc. So the problem of interpretation in that way is deferred horizontally throughout the system, and it doesn't work. An alternative interpretation is that inference happens at a higher computational or representational level. It's a bit reminiscent of the distinctions that David Marr made originally when distinguishing the hardware or implementation level in his book on vision versus the algorithmic and computational levels. And instead, what I'd like to propose here is more to say, well, there are multiple representational levels, of which the nature is not anatomical per se. It matters of what you can precisely encode at the lowest level in terms of features. Sensory features have to be combined into bigger wholes called perhaps objects, visual objects, and they eventually have to be combined at this higher level by comparison and correlation to other modalities. So the relationship of those levels, it's not about levels of performance, as Jean-Pierre talked about, but rather conceptual levels of representation, where the heart problem is basically considered a real problem. It's not something that one should deny, I think, but it is basically a problem of our imagination, because we've never learned to basically connect our phenomenal experience up there with lower levels of representation that are mediated more directly by neurons, or where neural activity is seen. So because our subjective experience is not structured that way, basically, one could argue. Now, for some experimental testing, I'll just take you through a couple of slides where the illustration is being done of how one could address, for instance, these cross-modal interactions, which are predicted by the theory. Here we see, very briefly, a two-photon imaging approach in mice that we used. Guido Meijer in the lab used this approach. Here's the mouse that is capable of behavioral responding, detection responses, no detections. It's sitting under a two-photon microscope, so this is the image you get from the V1 area, the primary visual cortex, but we also image in a higher visual area, AL. And what you see as green blobs here are basically the somata of neurons in layer 2-3 that are made fluorescent by a genetic calcium indicator called GCAM6. And this is the typical time series picture. You get 4 neurons in that whole ensemble with fluorescent responses that indicate basically somatic spiking activity. There's orientation selectivity here, and the colors represent different orientations of moving gratings presented, and the crosses here represent auditory stimuli that are added or are omitted. So this experiment was passive stimulation of auditory and visual stimuli to the mouse. One remarkable finding is indeed that you find auditory responses in mouse V1. That is, here is an intrinsic optical signaling image of V1 with the imaging planes here in colors. There are around 6 or 7% of visual neurons that respond even if there's only a tone. So without a visual stimulus, they will generate a significant fluorescence response. And this shows a lateral gradient with more cells towards the lateral border of V1. Next, we did an audiovisual detection task in mice, basically to get more at hits and misses, that is detection or no detection, seeing something or not. Indeed, this is the kind of psychometric curve I already showed, with a shift that you see based on the visual contrast, where the addition of an auditory stimulus will shift the curve towards the left. This means more accurate, lower threshold detection of the stimulus and a steeper curve. The converse also applies with audition. The addition of a visual stimulus to an auditory range of auditory amplitudes will give you a big shift and an increase in steepness. So these are significant effects. What you can then next do in mice is try to get closer to a possible neural correlate of consciousness, or at least of perception as measured by detection, which is to compose neurometric functions. So in addition to the sigmoid psychometrics, which is behavior that we saw, you can look at the individual neurons and say, well, they also have a sensitivity to visual contrast or auditory contrast. What is striking here between the areas, that is both low-level V1 and higher visual cortex in the mouse AL, is that it's quite well possible to decode whether there was a stimulus or not from the population response of all these 100 neurons or so in a session. And what you see over here is that these neurometric curves add up in a Bayesian decoding population approach, such that the AL population activity, so the higher visual cortex, closely matches the behavioral curve as you observe it by the behavior, the animal's behavior, detection or no detection responses, whereas V1 activity is not that closely matching the curve. So this higher area appears to be closer to the perceptual behavior and the perceptual decisions of the animal. This is all significant. Also here we find cross-modal interactions in the way of having both decreases if, let's say, you have a visually responsive neuron, that response will decrease if an auditory stimulus is added. The converse also occurs where there's an enhancement by the addition of an auditory stimulus. You basically see neurons out here that will only be modulated cross-modally at full contrast. Others will show the effect at the perceptual threshold. So in that sense, the lower and visual area do not differ very much. What you see in this lower graph is how the V1 neurons basically are either modulated by audition, by an auditory stimulus at full contrast modulation. These are the black dots, and the gray dots are when they are modulated at around perceptual thresholds, so halfway the psychometric curve. Same picture is seen for AL, and the overlap between these differently modulated populations is only slight. Finally, if we look at differences between seeing and not seeing in audiovisual trials, what you see over here is that now we start looking at hit to miss differences, so different types of trials, and you basically see that there's a positive difference, so hit trials tend to be having a stronger fluorescent response than the miss trials, both V1 and AL. For audiovisual, you see a slightly stronger response in AL, the higher visual area. And now we can also compose ROC curves, receiver-operator characteristic curves, where you see that if there's a positive bending curve, there is a detection capability in that neuron to distinguish hits from miss trials. And that also goes for particular neurons that do this for audiovisual conditions. This is only visual conditions. If we use these neurons and we average them together, it turns out that, again, area AL, the higher area, has a greater detection probability, but only in the audiovisual trial, so the cross-modal condition, whereas V1 doesn't have that distinction. Okay, then finally, in the last time period, I'd like to tell you a little bit about our computational approach to basically problems of consciousness and perception that are being pursued also in the context of the Human Brain Project. And this initial approach, I would say, it's not to build directly a model of consciousness that we could argue for as such, but it is to mimic certain interesting properties like inference, but also having generative properties, that is going the other way from a high-level representation back to the input. And I suspect also Karl Friston might talk about this later. The framework we work in here is predictive coding, and what we did, building on earlier work by Friston, but also Mumford, for instance, Rao and Ballard, is to say we're going to build a deep predictive coding network that is analogous to deep learning, but this time we do it unsupervised. We don't use backpropagation or another artificial algorithm. We want to do it unsupervised and basically use local learning rules. So the idea being that this approaches certain aspects of consciousness which are interesting by modeling inference in the brain. That also could be non-conscious, but also generative properties like imagery, of going towards the generation of images at lower levels of the system. Very briefly, the results here are that in this 5-layer example, so in this deep predictive coding network, Shirindora, postdoc in the lab, trained the network on 1,000 images. The generative model is able to reconstruct the images indeed, even if the network is trained on, let's say, ships versus horses, for instance. The presentation of novel images will lead to capable reconstruction, so you can basically take the high-level cause, the thing that is to be explained from the sensory input is being reconstructed. So this represents a triggering of the cause in a deep layer, layer 5, and it leads to reconstruction of the original input that the network has never seen before. Here we see some, let's say, neuronal tuning properties, where you see how the superficial layers of the model, layer 1, comparable to V1, successively down to deeper layers, leads to a greater complexity of the optimal stimuli. And this can be likened to the more complex properties of inferotemporal neurons, for instance, in responding to complex objects. So this is where I'd like to stop. In summary, I've shown you a couple of theoretical considerations that make a case for consciousness in certain animal species, especially emphasizing the organism's representational needs, like does it actually need a conscious representation to enable it to perform planned behaviors. I've argued for the importance of having a multimodal, immersive survey. I've emphasized that these multimodal interactions do occur, that V1, for instance, is not purely unimodal, and that there are functional differences between higher and lower areas also in the mouse. And then I would argue that, yeah, the inferential or predictive coding account is really becoming a very serious party in the game about explaining consciousness. I haven't addressed all aspects that are relevant, but basically I think there are a couple of arguments to say this, and that we need more comparison and testing of theories as well as theory development. Thanks for your attention.
Thanks very much, Cyriel. We have time for a couple of questions.
Here. So, yeah, I read a couple of papers some years ago by Krishna Sathian, arguing that some of, maybe a lot of, what is usually called the visual cortex is actually multimodal spatial cortex. It's actually, and is shared among a number of modalities. And the evidence were things like, occipital damage that causes tactile problems in tactile perception, and there are lots of. So I'm just wondering how much of your evidence for auditory for links between auditory and visual modalities is due to this fact, if it is a fact, that a lot of what it's called visual cortex isn't really visual, specialized for vision, right. It's specialized for space.
Yeah, so on the one hand, I think there's a lot of evidence now to say that indeed there are so many inputs into the visual cortex that it's better to talk about the whole cortex as a big associative structure. However, with, of course, still these specific inputs from the LGN that give a certain predominance to visual information. The second point about space, I think, is also highly relevant. I'd like to mention There's work, for instance, from Pieter Roelfsema, but also others, that there's working memory being represented in V1 activity also. There's also reward activity or reward predictive activity going on, as some of our own studies show, but also Mark Baer and Schuler have shown this. So real reward expectancy in V1 neurons in relation to learned visual tasks. And the point of space is highly relevant, because now what we find in area V1 and V2 of the rat is something like play cells, as you would normally find in the hippocampus. These are rats that walk around a maze, like a figure 8 maze or so, and these cells will only fire at particular corners, etc. And it's kind of hard to explain that only from the visual features, which would define those corners. So if there are symmetric situations, like a corner here that looks the same as the corner here, and the animal turning the same way, you still find a difference in responses. I think there's confirmative evidence from the Carandini lab also on this. So we really start to think more of the visual cortex also as predicting what's going to happen or what is currently going on, like predicting the present, both in a visual but also spatial sense. Yeah, so.
Yeah, you said that the hallmark of consciousness is that it's multimodal. Mm-hmm. But this is necessarily so. I mean, if I'm in horrible pain, I forget everything else. And it's your prediction then, if it remove all senses except one sensory modality, then you become unconscious. I think it's unlikely.
I absolutely agree that conscious experience can be dominated by one sense, like excruciating pain. However, going back to the example of the hand retraction from the gas flame, I would also argue that you're still in a situation, and you know this when you are suffering with pain, whether you're lying in bed or at the gas stove, and that this process could subserve learning or adaptation. So it would enable us to not only feel the pain, but also to say, well, what led up to this pain in this situation, and what could I do next time about it? But it's true. I think the theoretical argument to say that it's not only one sense, it relates actually to a thought experiment that Ned Block published in Ticks a number of years ago, which is called the consciousness in a bottle. And this is trying to imagine what it's like to have only one input, like V1 plus empty V5, like motion area. And I would indeed argue that it's not possible to have consciousness in a bottle, because you always have to interpret it as being about something, that is to identify the information as being about vision. And that could only happen if you have other types of information with which you compare and integrate. Yeah.
Okay, I hear there is a question on that side. Is that right? Yes, please go ahead.
I heard we have some hemi-neglect in the room. That is, one side of the audience tends to be...
Yeah, I checked. I didn't see any arms. Please go ahead.
Yeah, thank you. So thank you for the really interesting talk and the synthesis of a lot of different fields and perspectives. And I really like that you mentioned David Marr and his tripartite distinction. And I personally would say that it's actually more about the algorithmic level, the representations, and that you can see human cognition and consciousness maybe as an explosion of the relevance or the complexity within this algorithmic level. But my actual question is, what is your intuition or your perspective on how you can describe or explain consciousness given these three different levels of description?
Right, yeah. So the third level, I think David Marr distinguishes, is he calls it the computational level, but it's more about the goals of the theory and the strategy and so on. I tend to merge that a little bit with the algorithms because it's very much intertwined. Yeah, on the one hand, so my argument would be to expand, let's say, the computational or representational levels of what kind of information can you stuff into one level. And you can also see this, I think I have a slide on it, but it disappears, as having a network that could span from V1 to V2, V4, V5, that is about predicting one singular feature, a sort of low-level hypothesis about one feature. Whereas if you then try to combine all these hypotheses about single features, you get to the next level of combining the features into a visual object. So in that way, you could describe it as an algorithmic incremental approach. At some point, however, you end up with phenomenal experience, which is also not numerical, at least. It's more about quality. So in the end, I think it also has a non-algorithmic aspect. not, however, denying that there will be computations sort of underneath, so to say. Yeah, that would be my take on that. Yeah, thank you.
So now we will have Catherine Tallon-Baudry. I'm sorry that we delayed your talk. Catherine is a senior researcher at CNRS at the École Normale Supérieure in Paris, and she's done very relevant work on the neurophysiology of perception, visual perception, and today she will speak about visceral inputs, brain dynamics, and subjectivity. Thanks very much.
Okay, thanks Marie, thanks for the invitation. So I think we've heard so far many different accounts of what it could mean to be conscious, and we still have no clear answer regarding that, but clearly the link with intelligence has been underlined quite a lot, and my whole talk will be to consider that we should maybe shift our focus from things that are more related to cognition, as we've been doing so far in human brain research on consciousness, and more about subjectivity. Because indeed, when speaking about consciousness, we can consider two very different aspects. One which is related to the classical cognitive functions that obviously we can perform better when we are conscious. So this has something to do with consciousness. And there is another aspect, which is subjectivity. So cognitive functions, that's something we know how to deal with: attention, memory, language, control. Obviously, there are links between those functions and consciousness. And in this view, consciousness is more like a super cognitive function that helps me doing things in a clever way. Subjectivity is more difficult to define. We've heard this morning that sometimes we are lacking words to describe it properly. So I'm borrowing words from philosophers, but it has to do with the fact that when we are conscious, we do have a subjective experience that is expressed from our own first-person perspective. So it's related to the ideas of qualia, of the what is it like, to the heart problem, to phenomenal consciousness. But maybe to give a more intuitive version of this, I'm not a philosopher, is that if you're sitting outside, you're lying in the grass, you're looking just at the sun going through the foliage, you're perfectly conscious, you're having a very enjoyable and pleasurable conscious experience that's not absolutely not cognitively demanding. And that's really what I'm meaning, that I think it's time to shift our work toward subjectivity itself, which is the fact that consciousness, it does something to me, or more precisely, to have consciousness, it requires an experiencing subject. So for a long time, I think the experimental work that has been done in cognitive neuroscience was performed with this idea that if we understand well those aspects, at some point that will give us some clue about how subjectivity is being implemented. And I would like to devote the first part of my talk to explain why I think this is incorrect, before going into what could be a mechanism to generate subjectivity. So the initial proposals, such as the global workspace, for instance, were that attention would play a key role to generate a subjective experience. So very roughly speaking, the idea is that you have the bulk of unconscious information processing, and that to reach the top of the iceberg, that would be conscious processing. Attention is the mechanism that selects and amplifies information. So that would be the practical implementation to move things up from the unconscious to the conscious. And if we formulate things in a box and arrow manner, it would be that any visual information is first amplified, then you become conscious, then you decide to report, I've seen the stimulus in an experimental setting. So we wanted to test this simple assumption that surprisingly had not been tested as such. And in a very simple paradigm where people fixate, then they are cued to pay attention to one side or the other of the screen, and then in most cases the stimulus appears on the attended side and sometimes on the unattended side. And the only twist to the story that we use very faint gradings that people could only perceive about half of the time. So now we had a full design where the same physical stimulus could be seen or unseen, but it could be attended or unattended. So what we found looking at activity from the human brain using magnetoencephalography, and in particular from the, directly from the visual system, is that we have a full dissociation. So if we look at what distinguishes stimuli that are seen versus those that are not seen, this is the time the stimulus is being presented and the frequency decomposition of brain activity. We find clearly that there is a specific frequency band in the gamma range that correlates nicely with the verbal reports of participants. But interestingly, this is completely different from the signature of attended versus unattended stimuli. And there was no interaction between the two. So in this particular experiment, we could show that spatial attention and visual consciousness have completely independent neural correlates. So at least part of their correlates are completely independent. And this is something we could replicate several times. And so, whenever you see this kind of red box, it means across, shifting from Mac to PC generated a few issues. So it suggests that this gateway hypothesis is not correct, because that would clearly have been the prediction that you have to amplify so that any neural correlate of consciousness should depend on attention. Rather, it seems that we have two different mechanisms that are combined in the final decision of the subject to report I've seen the stimulus or not, which is always the difficulty we have, that we are going through verbal reports of participants. So this view also makes prediction, which is that there should be a lot of unexpected interactions between attention and consciousness, even at the behavioral level, which is indeed what we found. We found that depending on whether you're conscious or not of the stimulus, you deploy different type of attentions, which obviously could not be predicted from this type of perspective. And we also found the other way around with Claire Sergent that attention can trigger a conscious percept long after the stimulus is gone, which also does not really fit with a simple gateway hypothesis. So there are other popular ideas that we get a subjective experience because it's related to processing depth in general. I think now we have a lot of experimental evidence for unconscious processing, unconscious memory, unconscious semantic processing, unconscious control. So all those functions that are supposed to be particularly well developed in humans or exclusively humans, and that should be related to subjectivity, actually do not really explain subjectivity as such. So it's certainly time to reconsider subjectivity, but before suggesting a neural mechanism to account for it, I would suggest, I want to go through one more thing that has been argued, that subjectivity simply does not exist, that it's something which is an illusion, or at least it's something which is nothing else than a cognitive construct, and that we don't have to resort to any specific mechanism, dedicated mechanism to account for it. So this idea is rooted in the in examples like when you see a visual scene, you have a rich visual perception. Could I get the movie running up there? Thanks. Okay, it's not working, sorry, forget it. The change blindness experiment, so here it bugs, but typically you're flashed with a scene and you can't pick out what is being changed. Here you can pick it up because the movie is not working properly. And this has been used as an argument that although you have this feeling that you can see it all, you are not able to report the details, so it means that your rich experience is just an illusion. And this can actually be quite easily accounted by taking into account how the visual system is built. So here I'm just redrawing a model from Hofstein and Aissa, which has been a very influential model derived from perceptual learning, so not particularly from consciousness studies, with the idea that the visual system extracts features and combines them into simple shapes up to the point of scene recognition. And this is done very quickly in a feed-forward sweep of implicit, that I would relabel unconscious processing, to reach those higher order visual areas where you would get an initial vision at a glance. And to retrieve details, you would have to go down backwards toward this visual hierarchy, what they call vision with scrutiny, to retrieve the original details that were used to build the scene. And if we reformulate that in terms of consciousness, it means that the initial locus of conscious experience should be here, and that you would retrieve conscious details in only an optional manner, and that would take more time. And that can explain easily change blindness experiments, and we could actually verify that all the predictions derived from this model hold true using new hierarchical stimuli. We can show that if you ask people either to respond regarding global content or local details, they are always faster for the global content. If you decode brain activity, when people are asked to perform at the local level, you nevertheless decode the global activity, which shows that people have to go through this stage. It's a mandatory stage. We can't escape it, even if it's not task relevant. And if we ask people to spontaneously report what they are seeing, they always report the global level. So this suggests that we cannot dismiss subjective experience as a mere illusion, and so we have seriously to reconsider what does it mean that it requires an experiencing subject. And if I reduce that to the idea of building first-person perspective, the question now becomes, how can we build first-person perspective? So you could think it's trivial, because if anything's happening in my brain, it has to be from my perspective. That's true, it can't be your perspective. If there is a perspective, it's going to be mine. But it doesn't mean that it's trivial that there is perspective at all. And that's, of course, something we cannot experience by definition, but that's something we know as scientists. There is unconscious processing, and by definition, this unconscious processing is not equipped with first-person perspective. So what it means is that processing information with first-person perspective, that's not the default mode of processing of the brain. So yes, there is something that we need to explain. So the proposal that I'm making is that to account for first-person perspective, what we need is to have a subject-centered reference frame from which first-person perspective can be expressed. And this ego-centered reference frame would be derived by the neural monitoring of visceral organs. So why visceral organs are particularly interesting? Both the heart and the gastrointestinal tract actually contain cells that act as pacemakers, so that if you disconnect them from a brain, they go on doing whatever they're doing. So the heart goes on beating, the stomach goes on contracting, etc., etc. So it means that they are constantly feeding the brain with ascending information about the fact that there is an organism around the brain, which is a very simple and simple way for the brain to generate something that says, yes, there is a whole organism, and that's from this reference frame that first-person perspective can develop. So it means that whenever, for instance, visual information is being processed, if it interacts with this ego-centered reference frame, then you can derive a subjective experience. And I think that's what essentially is different from computerized version of artificial vision, for instance, because a computer that is doing battery monitoring, for instance, which would be the equivalent of monitoring visual inputs, for instance, it doesn't change its outputs depending on the level of the battery. These are just completely separate modules. But in a living organism, it makes a lot of sense that this should not be separate modules. And unconscious processing in this particular case then could be either a reduced ego-centered reference frame or an absence of connection between, for instance, visual processing and the self-centered reference frame. So this is, of course, not limited to visual perception. This is something that we could find in a lot of other cognitive domains, such as, of course, self-consciousness, but also emotions, which are highly subjective feelings, or decision based on subjective preferences. So this is certainly not the first time suggesting that bodily inputs might matter for consciousness in general, and very often feedback from action, for instance, has been considered as important. Vestibular inputs also certainly play a role. I'm certainly not suggesting that these are two completely opposite views. Rather, they probably converge at some point. But still, I mean, if you stay completely still, you just have a little bit of vestibular inputs. Or in locked-in syndrome patients who are fully paralyzed, there is no action by definition, and they are nevertheless perfectly conscious. So I think that life-sustaining organs, such as the heart and the gastrointestinal tract, are good candidates to provide a very stable source of bodily information that might matter for consciousness. Oops. Okay. So just 2 words briefly. In this view, there's no need for the bodily state to change, so I'm not talking about changes in heart rate or digestions or things like this. What matters is that there is some upcoming signal. The second important thing is that I'm not talking about conscious experience of your visceral inputs. You might sometimes feel that your heart is beating faster, but that's not something very common. And that's really not what I will be talking about here. I'm proposing something which is purely mechanistic. What is being experienced is the final output of all this neural processing. One last word is that visual information is potentially very widely available in humans because it targets quite a number of cortical regions through many different pathways. So it's a system which is extremely redundant in a way. Okay, so far for the general hypothesis, how can we test it? So I guess you're all familiar with what a visual evoked response is. You present a visual stimulus in humans. If we measure non-invasively, we get very noisy data. We do it several times, average, and then we get a transient response. We can essentially do exactly the same thing using the heartbeat as a stimulus. And it repeats naturally, and if you average, you get a transient response. Okay, these are just simulations, these are not real data, and this is something which has been known since the 80s, thanks in particular to the labs of Chandri. One example from this paper by Kern and collaborators of an intracranial recording over the primary somatosensory cortex in humans, locking the data to the heartbeat, you get a nice transient response with a certain latency, which is nicely focal over the primary somatosensory cortex. So we can use this measure to probe this hypothesis, and we began by probing subjective vision, so vision at threshold, where typically the hallmark of those experiments is to have the subject reporting I see the stimulus. So here the idea is that in this statement, of course, there are things which are related to visual processing, and that's the part in the sentence that would correspond to the stimulus itself. But the proposal is that we also have some processes which are related to the first-person experience, and that might be related, that might be indexed by neural responses to heartbeats. So in a way, the idea is that we have spontaneous fluctuations in this reference frame, and if a weak incoming stimulus arrives at different moments in this reference frame, it can be perceived when the frame is large, or it can be missed if the frame is small. So that's exactly what we did. People fixated, then after a while, when they fixated properly, the fixation point turned red, and then a faint grating could appear or not, and then after another delay, they just had to say whether they saw something or not. Okay, so it was all titrated, and we looked, so we have hits and misses, and we looked just before stimulus onset during this warning period at neural responses to heartbeats using MEG in humans. And what we found is that indeed, we do have larger responses to heartbeats in hits, interestingly also in false alarms. And this occurs in regions that are not often associated with visual processing, in the ventromedial prefrontal cortex and right inferior parietal lobules, which are regions of the default network. And I'll just go through a number of the controls we did. So alternative interpretation, is it attention inside, outside? It doesn't work well, because if it was a balance between attention inside and outside, it would predict that if you are paying attention inside, you get larger responses to heartbeats. This is known. But then you should miss the stimulus, and we observe exactly the opposite pattern. There's no change in cardiorespiratory activity. It's not the heart is beating faster, that you are breathing faster or whatever. There's no massive change, neither in bodily state nor in brain state. There's not more or less alpha power, for instance, or things like this. So just briefly, I think whatever those phenomena are, they cannot be dismissed easily. If we talk a little bit about effect sizes, which is something important, so here what I plot is the amplitude of the response to heartbeats in one participant. So if it's small, medium, or large, and whether in those trials the participant was responding less often I've seen the stimulus as on average or more often. And if we do that in all participants, first you can see that the effect is robust in all participants but 2. We see the effect, and that we can explain about 10 points in hit rate using the heartbeat evoked response. So this is as large as any classical effects described in electrophysiology, like for instance, attentional effects on visual perception typically explain this type of differences in hit rate. So whatever it is, it cannot be dismissed as being a marginal effect. The other thing we investigated is what is the nature of the information that is brought by this neural response to heartbeats. And for that, we went to the simple model of signal detection theory, where vision at threshold is all about distinguishing between noise in the system from information, signal about the presence of a stimulus. And the separation between those two curves indexes perceptual sensitivity. So of course, if you have more information, you have a better perceptual sensitivity. There is another way the hit rate could change, is that if people change their decision criterion. So we could tease apart those two parameters of the decision, and what we find is that the heartbeat evoked responses is actually changing the sensitivity. So it means that you have more information on which to decide whether a stimulus was present or not. So it means that the neural response to heartbeats behaves as sensory evidence in the final decision I've seen the stimulus. Obviously, it does not carry directly visual information, but it might carry information about the subject who is experiencing the stimulus. Okay, so we do have some evidence that neural responses to heartbeats have something to do with subjective vision. We also investigated more directly whether they have something to do with perceived self-relatedness, which is something not very easy to study properly. So we went for an interrupted thought paradigm. I'll just first explain how it goes before explaining why we went for this. So it's just the super boring experiment for participants. They fixate a gray screen for a long period of times. And then from time to time, this visual stimulus appears, in which case they have to remember what they were thinking about, okay? Because they get bored, they start thinking about different things. And we asked them to rate the self-relevance of the thoughts that they were in at the moment this visual stimulus appeared. So if they did not know, they could skip. And we rated self-relevance in 2 different ways. The most classical way when we think about self-relevance is whether you were thinking about yourself. So that if you're thinking, oh, I'm thirsty, or I'm jet lagged, or things like this, then you're thinking about yourself. But if you're thinking, oh, tonight I have to go to the supermarket, actually you're thinking more about your grocery list, but you're the agent in the thought. So you are also present as an agent. If you're just thinking it's raining, well, not in Barcelona, but in Paris, you would think it's raining, that's more neutral. I mean, of course it's your thought, but you're not very much engaged as an agent in this thought. So we asked participants to rate those 2 aspects of the self that we labeled the I, the agent, and the me, the more reflective, what you're thinking about, and rated each trial according to self-relevance along those 2 scales, while, of course, we measured the electrocardiogram and magnetoencephalographic activity. And what we found is, again, that in the ventromedial prefrontal cortex, neural responses to heartbeats distinguish between thoughts that are highly self-relevant or less self-relevant along this reflexive dimension of the me, and in the posterior cingulate cortex along the agentive dimension of the self. This is a result we were lucky enough to replicate in intracranial recordings in epileptic patients at the Hôpital Pitié-Salpêtrière in Paris, where we had one patient with an electrode in the PCC and another one with an electrode in the VMPFC. And here we could go to a real single response to heartbeats and single thought correlation. And it works quite well. So here what you see plotted is here the scores that the patient gave about the thoughts, and here it's the normalized amplitude of the heartbeat evoked response in this patient. And you can see that we can get quite nice correlations between the two, even if those are single events. So, okay. Again, we run all kinds of controls. I won't go into much the details, but there's nothing obvious in terms of cardiac electrical activity, heart rate variability, respiration rate, etc., etc. No massive change in bodily state or in brain excitability, and the effects are truly locked to heartbeats. So it's just a starting point, but it seems that those neural responses to heartbeats might have something to do with how we can generate first-person perspective, so how we can generate an experiencing subject, at least one of the important building blocks of an experiencing subject. There is also other evidence that I will not present, so some work done by my former PhD student, now a postdoc in Olaf Blanke's lab, about bodily self-consciousness, where also they have larger neural responses to heartbeats in those nice settings they have developed. And also other evidence from my lab that I will not present because of time constraints about self versus other distinction and subjective values in decision making. So to conclude, what I suggest is to shift from classical information processing in the brain that can be easily modeled in an artificial system to the interaction between classical information processing and first-person perspective. However, whatever the way first-person perspective is generated, here we are proposing one mechanism that could be interesting. If this hypothesis turns out to be true, I mean, we have good evidence that it might be true, but we are certainly not sure yet. It suggests that subjectivity obviously requires a brain. I'm certainly not saying that you generate consciousness in the heart or in the stomach, but it's still an organism property. And I think it's important to ask, what is the right description level for subjectivity? It doesn't mean that we cannot study the mechanisms that generate them, but are we so sure that it's enough to have a brain to generate subjectivity? I honestly don't know, but when I read about the reports about this group at MIT that is trying to keep a brain alive in a vat, I think it's probably time that we really ask this question. Okay, on this, I will thank those who did the real work, and in particular, Yongdong Park and Mariana Babo-Rebello, who were very brave and embarked on those initial studies on the neural responses to heartbeats. And thank you all for your attention.
Thanks. That was a terrific talk. So I have two very short questions. One is there a heart rate effect on the way the stimulus looks? So I can imagine investigating this using a two-interval forced choice procedure where you combine that with the kind of technique that Marissa Carrasco uses to show that attention affects appearance. So that's one question. The second question is this effect mediated by attention?
So first question, what we show is not a cardiac cycle effect. So it's not whether the stimulus comes close to a heartbeat or far away from a heartbeat. It's really how the brain is responding to the heartbeat. So we tested that because there are some evidence in the literature that Depending on where the stimulus appears with respect to cardiac phase, that might have an impact on how it's processed. I think the best evidence is for fearful or fear-related stimuli. In our hands, there is absolutely no impact of the cardiac cycle. Okay? So what we have is a distinction between seen and unseen. And I don't really see what we could test in terms of the different quality that it could have. Okay, so that's, I know it's always confusing, but there are those two things in the literature: responses to heartbeat, neural responses to heartbeats on the one hand, and cardiac cycle effects on the other hand. Cardiac cycle effects are quite specific to fearful stimuli. There might be other cases where they are important, but there are a lot of conflicting evidence for the other cases. And in our hands, it has no effect, except sometimes on reaction times, but not on the whether you've seen something or not.
Instead of seen versus unseen, you could make the stimuli. Instead of seen versus unseen, you can make the stimuli stronger and ask whether the first one has more contrast than the second one, or.
Yeah, I know the Carrasco experiment, but then we run into those issues of time. If you present things in a sequence, then what you're looking at just before the first stimulus onset is likely to impact the first one rather than the second one. And we can't really measure the neural responses to heartbeats during stimulus processing, because then we mix two different things. I mean, we could, but it's not very clean. Yeah. And regarding your second question, is it attention? Yes, of course, that's always the question. So as I said, for the experiment on vision at threshold, it seems unlikely in the sense that the attentional interpretation would give the opposite results than what we have. When it comes to spontaneous thoughts, yes, of course, there is something related to attention. I mean, if I'm thinking about net block, I'm paying attention to net block. It comes together. So in this experiment, we just can't distinguish between the two. The fact that we can distinguish between the I and the me suggests that it's not only attention, because if you think I'm going to the supermarket, you're paying attention to the supermarket aspect more than to the fact that it's you who is going to go there, except if you're going instead of someone else, etc. But that's why it was the participants who did the ratings of their own thoughts, and we trained them quite a long time to make sure that they got what we were meaning. Yeah.
Next question, please.
Hello, here.
Okay.
Okay, since we are talking about subjective experience, there is a theory on emotions that says that when, let's say, responses from different systems synchronize, for example, responses from like visceral responses and then responses from the brain, then this is when the emotional processes happen. Do you think that something like that can apply as well to identify this subjective experience so as to link the brain output with the visceral responses?
Yeah. So, 2 points here. The first thing is that emotions have a very subjective component, which is not often studied as such, I mean, surprisingly. And the second thing is that here, actually, I never talk about visceral responses. So I think that the main difference with the emotion literature, where there is this ongoing debate, which is, okay, when you feel an emotion, then you might have changes in your heart rate, for instance, and what caused what. Here, I've really emphasized the fact that there are no changes in heart rate at the moment where we measure the differences in how the brain is responding to those inputs. So it's a loop, of course, but we are looking more at the ascending part of the loop, while very often when people measure the changes in the body, the information has been processed by the brain already, yeah? Because, I mean, your heart doesn't change by virtue of perceiving directly the stimuli. It's mediated by the brain. So people often look at the net output of this full loop while we are trying to isolate the ascending part. So that's, I think, the main difference at the moment. But of course, there are links between those. Yeah.
Okay, 2 more questions.
Oh, okay, thank you very much. Here. I was thinking about people with some sort of arrhythmia, cardiac arrhythmia, or some syndrome like Wolff-Parkinson-White or something like that. Do you think that people with arrhythmia may have a different subjective experience in this sort of experiment?
So again, I don't think it's the structure of the incoming input that matters. What matters is the fact that there is an input. And that's where the hypothesis is really just an hypothesis, because it's not easy to falsify. You can't easily remove the heart of a human participant. I don't think our ethical review board would accept that. And we would not have a living brain to record from. So that's the limit of this hypothesis, and that's why I think we need a lot of experimental evidence before we can make a clearly convincing case, because it's not easy to falsify. But we try to keep the assumptions minimal, and the minimal assumption is just that you need some input. No matter how it's structured, this is what is, that's what matters, is the fact that there are some inputs. And then the second thing is that here, okay, I presented everything about the heart because we can quite easy, well, it's not so easy, but we can measure the neural response to the heartbeat. So we have a good index there. But as I've said, that's certainly not the only interesting organ. The whole gastrointestinal tract is interesting as well. And as I mentioned in the introduction, probably this combines also with vestibular and proprioceptive inputs. So I don't think it's unique to the heart either, which would make sense in a way, that it does not rely on just one specific connection. Yeah.
So what is the relation between the heartbeat and the alpha oscillation? Because yes, it's a classic question.
Yeah, no, it's a very good question. So the short answer is we don't know. And if I develop a little bit, that it's not so easy to know if we want to do it properly. Because I skipped all the boring details, but we do have a huge stimulation artifact in a way in all those studies, in the sense that whenever you record brain activity, you also record the electrocardiogram at the same time. So when you average locked to the heartbeats, you get both brain data and cardiac data directly. So we have all kinds of controls and tricks to see that, but it means that it's not easy to analyze properly any oscillatory activity, knowing that you more or less have a Dirac in the data, in the middle of the data. So we can attenuate, but still it's there. We thought moving intracranially would help, but then we get not only the cardiac artifact, but also the pulse-related artifact. So it's not so easy to do it properly. I do have the feeling that it has something to do with it, but I'm not sure. What we did, but that I did not present for the sake of time, and because it's more related to brain dynamics and not yet to any behavioral consequences that we can measure, is the link between the stomach slow rhythm. So it's a very, very slow rhythm, 20-second period. And here we showed that the frequency band that is really coupled to this rhythm is the alpha rhythm in the parieto-occipital sulcus, essentially. So there is probably a link, but at the moment we don't know more, and for the heart it's a bit tricky to get into it.
Okay, thanks very much, Catherine.